So our next speaker is Sally Till, and she's going to tell us about rat models of SYNGAP. Now, mice are relatively smart, but rats are smarter. So a lot of people are turning to using rat models for many of these studies, and I'm sure Sally will tell us more about that. And Sally's from the University of Edinburgh.
Great, thanks. I'm Sally. I'm a research fellow in the KIND Lab, and we are part of the Simons Initiative for the Developing Brain, which is a virtual institute at the University of Edinburgh. And the main aim of SIDB is to begin to understand some of the biological pathways that underlie different types of monogenic neurodevelopmental disorders, with the hope that we can use that information to deliver rational therapeutic interventions. So today I'm going to tell you about some of the work that we've been doing on SYNGAP1, and we've been modeling this, as Rick mentioned, in rats. So I thought I'd just spend a moment telling you about rats, because although they look very similar to mice, they are not just big mice. And it turns out, actually, that they are less evolutionarily similar to each other than we are to chimpanzees. So whereas humans and chimpanzees separated evolutionarily between 8 and 10 million years ago, rats and mice separated more than 12 million years ago. And we think that rats are a really interesting model organism, in part because they have a really rich behavioral repertoire. So if you've ever known a rat, you know that they're really crafty. They're highly flexible in novel situations. And they're really a sociable species, and this comes in handy for them because unlike mice, which exist in sort of territorial small groups, rats tend to live in these really big communities. So social skills are a really important behavior for them. And we're particularly interested in these two cognitive domains because they are specifically affected in many neurodevelopmental disorders, for example, SYNGAP1 and autism. So rats also have larger brains, which electrophysiologists like because they can stick more electrodes into them, for example. The drug metabolism and pharmacokinetics in humans is more similar to that in rats than it is in mice and other model organisms, lower model organisms. And because of recent technological advances over the last decade or so, it's become pretty straightforward to make genetically engineered rat models. So this is not to say that we think that rats are a better model species, but we think that they really complement existing model species, and we would really encourage everybody to come together and to try and solve these different mechanisms underlying these neurodevelopmental conditions. Hopefully that rats will be able to do this, help us do this. So at this point of the day, I don't really need to introduce SYNGAP1. And thank you to Gavin, who did a really nice introduction for me about all of the different isoforms of SYNGAP. And as he mentioned, although there are so many different mix and match isoforms, so any of these N-terminal isoforms can mix and match with these C-terminal isoforms, all of these isoforms, they share this common core domain. And really what I want you to take home from this picture, this rainbow-colored picture, is all of these different functional domains in SYNGAP. And as Gavin mentioned, individual isoforms have these very different functions. So interestingly, most of the pathogenic variants in SYNGAP1 are predicted to lead to premature termination. And a lot of times this is predicted to cause instability and loss of one copy of SYNGAP. And so for this reason, many SYNGAP1 individuals are thought to be haploinsufficient for SYNGAP1, so half of SYNGAP. But there are several individuals with mutations that are not premature stops, that they're a missense mutation which changes the identity of a single amino acid, and the rest of the protein remains the same. And at least 14 individuals exist with missense mutations in the C2 and GAP domain. And this suggests then that these particular domains, which regulate the GTPase activity of RAS and RAP, are particularly important for the clinical traits associated with SYNGAP1 haploinsufficiency. So to test this hypothesis that it's specifically this enzymatic function that is quite important, we have selectively targeted the deletion of these regions, and we've used a technique, a special genetic technique called zinc finger nucleases that targets exons 8 through 12, and the regions are shown here. And down below, I've shown you an alignment, the top line of which is a normal copy of SYNGAP, just in this region. And each of these individual letters stands for a single amino acid, so a single building block of this protein. And down below, we have this mutant protein, which we call delta gap right here. And wherever there's a dash, this means that one of these amino acids is missing. So you can see that these mutant animals still have normal SYNGAP before and after this deletion, but they're missing what turns out to be 377 amino acids here. So importantly, what we've been able to find is that this mutant SYNGAP, which is shown here in this blot, this plus delta gap lane here, a smaller protein runs lower on this particular type of gel, which is called a Western blot. But we find if we look for it in protein extracts that are enriched in synapses, we can see that SYNGAP is making it to synapses. And we've done this in a second way, which I think I'll probably just gloss over, but we're able to identify that not only does it make it to synapses, it also interacts with really important glutamate receptors that it's known to be binding partners of. So heterozygous C2 gap deletion SYNGAP rats, that means rats that have only one copy of this particular mutation. They are healthy and they're viable, and they're largely indistinguishable from their wild-type littermates. But if an animal carries 2 copies of this mutation, it's lethal. So the animals are born, but they don't make it to more than 5 days of age. And this is consistent with findings in SYNGAP heterozygous and SYNGAP homozygous knockout mice as well, that this is a homozygous lethality. So for the remainder of the talk, what I'm going to talk to you about are these heterozygous SYNGAP C2 gap deletion rats, but I think for simplicity's sake, I'll probably just lapse into calling them SYNGAP mutants or gap deletion rats, okay? And what we've done is we've taken these animals and we've tried to look for symptoms, behavioral symptoms, electrophysiological symptoms, that may correlate to clinical traits associated with SYNGAP1 in individuals, humans. And the first of these that I'm going to tell you about, I noticed early one morning just watching the animals hanging out in their cages. And so what I'm going to show you is this video, and if you focus on this guy in the middle here, you're going to see some non-typical behavior. So he starts to nod his head quite a lot, and you'll see this, this behavior a few times. He's done it again here, and now when he rears up, the next time this clonic event will happen, it will spread to his forelimbs. It's just about to come. And he loses his balance. So this guy, all of these behaviors that he's been showing, he is actually one of the more severe on this spectrum, but I thought he was good because sometimes it's hard to get your eye into these types of behaviors, but all of these different behaviors map directly onto the Racine scale for seizure characterization, and these are happening spontaneously, so it's just something that happens. But to test whether or not these different behaviors actually did represent some sort of epilepsy, and these types of behaviors are associated with absence epilepsy in rats. We recorded from rats with chronically implanted 32-channel skull surface grid EEG probes. So here's a little cartoon of it. It looks a bit like a Christmas tree, and all of these little dots are individual electrodes. It's kind of like an EEG cap that you might put on a human. And I'm just highlighting here 10 of these channels in orange and in green. And each one of these channels lies over a different brain region, and these are labeled here. And what you can see from these squiggly lines is that there's this characteristic squiggly line over here, which generalizes across all of these electrodes. And these are called spike and wave discharges, which Gavin and we've heard about earlier today. And importantly, we find that these spike and wave discharges correlate with a signal in our accelerometer, which is when the animals are actually bobbing their heads. So when we quantified this, we found that a larger proportion of our gap deletion rats were showing these types of spike and wave discharges compared to their wild-type littermates and compared to wild types from other colonies that we maintain. And when we quantified the number of spike and wave discharges, or SWDs for short, we find more of them in Syngap mutant animals, shown in blue, than in their wild-type littermates. And so we wanted to know whether or not these might actually relate to absence epilepsy. And so to do this, we treated the animals with a drug called ethosuximide, which is a first-generation anti-epileptic, which is commonly prescribed for individuals with absence epilepsy. And we found that a single dose of ethosuximide did suppress these spike and wave discharges compared to untreated or saline-treated animals. So this suggests that these behaviors and this abnormal EEG that we're seeing does relate somehow to absence epilepsy. And importantly, absences are routinely seen in individuals with SYNGAP1. So one of the associative learning paradigms that we've used to begin to understand how the role of SYNGAP in cognitive function is called cued fear conditioning. And I'm just going to take a minute to explain to you what we're doing, because I'm going to talk about this for a little bit later on. So initially, after animals have explored sort of a neutral box on 2 non-consecutive days, we place them into a conditioning chamber which contains this electrified grid. And while they're in here, we give them 6 paired presentations of a conditioned stimulus. Now, a conditioned stimulus is a stimulus that would normally be neutral. And in this particular case, it's a flashing light. And this conditioned stimulus, the flashing light, it co-terminates with a mild foot shock. And the mild foot shock is kind of alarmingly tingly, and the animals find this really annoying. So 24 hours after conditioning, we put the animals back into this neutral box, and we look at their defensive behaviors in response to unreinforced, repeated presentations of this conditioned stimulus, so the flashing light. And here they don't receive any foot shock. We're just looking at their defensive responses. And in this case, what we're looking for is freezing behavior. And you've seen freezing behavior I'm sure, in a park somewhere when a squirrel notices a hawk fly by and they just stop. So freezing is technically defined as absence of movement except for breathing. So here we can see similar learning curves between wild-type animals, again shown in black, and the Syngap mutants in blue, such that they increase the amount of time that they spend freezing in response to these repeated presentations on the day of conditioning, compared to when they first encountered the box. And we've done a really important experiment, a control experiment, with animals who saw the flashing light but never had a foot shock. And we can see this entirely different behavioral profile here. And this suggests now that the flashing light is not aversive on its own. So that's on conditioning day. When we look at the recall test, what we can see in wild-type animals is a robust freezing in response to the first few presentations of this flashing light that gradually decreases with increasing number of presentations. But in contrast, the Syngap mutants show this elevated freezing, and this doesn't decrease over time. So this is actually a form of learning, this learning that yesterday, the flashing light predicted something bad, but today it doesn't, and this is called extinction learning, and it's not forgetting. It's a really important form of learning because being able to associate stimuli and situations with danger and safety is really critical for survival and for adaptive behaviors. And we know that adaptive behavior is one behavioral domain affected in many neurodevelopmental disorders. So we next tested whether blocking seizures could improve this impairment in extinction. And to do this, we used ethosuximide because we had seen that it blocked spike and wave discharges in this rat model. So in this experiment, we gave ethosuximide 1 hour prior to conditioning, and again 1 hour prior to recall. And what we found, here I'm showing you saline-treated animals, so that's a control, with filled circles, and ethosuximide-treated animals in open circles. We can see that ethosuximide didn't affect this behavioral profile on the day of learning. But interestingly, we see a significant improvement in the Syngap heterozygous mutants compared to saline alone. So this suggests not only that ethosuximide is improving it over trials, but overall, if we look at an index that we calculate that compares the freezing at the end of this recall test compared to the beginning, we see this is a significant difference. So because ethosuximide didn't have an effect on the day of conditioning, we predicted that there was a specific effect of ethosuximide on the day of recall testing. But another possible explanation for this is that ethosuximide instead just increases activity, and that this hyperactivity could sort of mask the freezing behavior and confound our analysis. So to rule this out, we took a separate group of animals and we gave them saline or ethosuximide 1 hour prior to looking at their locomotor activity in an open field. And so you can see this locomotor activity plotted over 20 minutes of time. And again, ethosuximide-treated animals are in the open circles, and saline are in the filled circles. And you can see that ethosuximide is not having an effect on activity, suggesting that this may be a cognitive improvement. So in these same animals, we waited 1 week before putting them through, again, this fear conditioning paradigm. And we waited 1 week because the half-life of ethosuximide is about 12 hours, and we wanted to make sure that it was well and truly out of their system. So in this experiment now, we're again giving 2 injections, but only one of them is containing ethosuximide, because what we want to do is to test whether or not ethosuximide is required on the day of recall. And what we found really surprised us is that if we gave ethosuximide specifically 1 hour prior to recall, we don't see this improvement in cognitive performance, whereas ethosuximide given specifically before conditioning did have an effect on recall, and we see an improvement in extinction here. So what's happening during this period between conditioning and recall? So newly learned information, in this instance that a flashing light predicts a foot shock, can be consolidated into long-lasting memories that can be accessed after a delay on the order of days or even years later. And there's a really large body of work that's shown that sleep is very important for consolidation. So we next asked whether sleep is disrupted in our SYNGAP rat model. So to do this, we quantified sleep and wake states over 24 hours, and we did this in animals that were implanted with these EEG probes. So here is a schematic of 24 hours of a rat's day, and it starts with 12 hours of lights on, and it ends with 12 hours of lights off. And because rats are nocturnal animals, they're asleep most of the time during the lights on, and they're awake most of the time in the lights off. So when we quantified the amount of REM sleep over this 24-hour period, we found that wild-type animals were showing much more REM sleep than the SYNGAP mutants, at least during the lights-on phase. And if we quantified this over the entire 24 hours, we found not only is there a decrease in the amount of REM, but there's a decrease in the average length of each REM episode in these SYNGAP mutant rats. And this seems to be specific for REM because we don't see a difference in NREM or in the time of the amount of time that the animals spend awake. And importantly, we find that if we give an acute dose of ethosuximide, the timing of which is shown here by this orange dotted line, we can increase the amount of REM sleep in our SYNGAP mutant animals, which are shown here in these orange-filled dots, at least during the light phase. So it appears then that ethosuximide improves seizures, REM sleep, and cognitive measures, but how these processes are interlinked at a mechanistic level really isn't known. But clearly it's important to understand because epilepsy and sleep disorders often co-occur with SYNGAP1 and other neurodevelopmental disorders such as autism. And how these contribute to cognitive phenotypes as seen in affected individuals is not well understood. And this is what our future studies are going to address. So all of the studies I've told you about until now have been using this heterozygous C2 gap deletion mutant SYNGAP model. And before I wrap up, I just want to show you some work that we've been doing to see whether or not this is unique to this particular type of mutation, because this mutation does not actually occur in any individuals. And to do this, we've compared these different types of phenotypes, these different symptoms, in rats that model SYNGAP haploinsufficiency. And so we've made a rat with a frame shift mutation in exon 8, which leads to a null allele. And we find that heterozygous knockout rats show approximately 50% expression of SYNGAP compared to wild types. And we find that homozygous knockouts, like our C2 gap deletion mutants, are also lethal. In these SYNGAP heterozygous knockouts, we find an increase in spike and wave discharges. We also see that these can be suppressed by ethosuximide, just as the C2 gap deletion rats are. And we see this enhanced fear phenotype, so that there is an absence, an impairment in fear extinction. So this suggests that This suggests then that both of these different SYNGAP rat models are showing a consistency, and that this enzymatic function of SYNGAP is quite important, at least for these particular phenotypes. And we've looked at many other phenotypes as well. We see a good correspondence of these different symptoms across our two models. And I just want to briefly mention this because it may be interesting to discuss at a later time today. We see our rats don't seem to behave in the way that we would expect them to. If you put an animal in a maze, you expect it to take certain approaches, and our SYNGAP rat models don't seem to be paying attention or interested in the same things that wild-type rats are. And this is just one example. It's a marble interaction task. You place the animal in an open arena, and it contains different marbles, and they're sort of the size that a soccer ball would be for us. And actually, wild-type animals treat them a little bit like soccer balls. They'll roll them around and interact with them. But the SYNGAP models are not interested, or not as interested anyway. And we've seen a similar type of symptom in other different types of behavioral paradigms. So it might be interesting to think about what this could represent. So in summary, I've shown you that the heterozygous loss of these enzymatic domains results in spontaneous seizures and impairment in fear extinction. The phenotypic similarity to SYNGAP heterozygous null rats suggests that this enzymatic function of SYNGAP is quite important. And it also gives us an insight into potential therapeutic strategies because it gives us a druggable target to try and intervene with this enzymatic function. I've also shown you some data that suggests that ethosuximide improves cognitive, sleep, and seizure phenotypes in these rats, which suggests a route to potential clinical benefit in humans. In its favor, ethosuximide has a really well-developed safety profile in adults and children. That said, we don't have any reason to suspect that the actual mechanism of action of ethosuximide is what is necessarily important here. It could just be the suppression of seizures. So going forward, what we're going to try to do is to try and understand whether the cognitive benefits that we're seeing from ethosuximide result from the seizure suppression in general, or just to the sleep improvement, or maybe it's involving all of those, both of those. We're hopefully going to look very soon into whether ethosuximide improves other cognitive phenotypes that we see in our SYNGAP rat models, and whether antiepileptics in general can be beneficial in other neurodevelopmental disorders. So this has been a really big group collaboration with our collaborators in Bangalore, India, and McGill University. Nicole, Denai, and Lisa were really helpful with the behavioral aspects of this. And Ingrid has done all of the EEG analysis and recordings in Alfredo Gonzalez-Sulcer's lab. So thank you to you all, and thank you to all of our funding partners as well.
All right, so we're going to take 2 questions. I'm going to bring everybody back for the questions after. So, did you guys do did you measure any oscillatory activity plus or minus ethosuximide?
No, we haven't done that yet.
My guess would be that one of the things that FSLXMI might be doing is improving, data algorithms or something like that, which is important for consolidation.
Yeah, be really good to look at.